My name, thank you, Sarah. I didn't mess up my name. I have a very, exotic name, Sergiy Borysov. Um, I'm actually, some people ask me where I'm from. Well, I am right now from nowhere, actually. I used to teach at University of Tampa, and now I'm transitioning to St. Leo University, so technically I'm nobody's, except I belong a little bit to Moffitt Cancer Center. So we're gonna talk, well, let me say that. I did one PhD in Ukraine, and then I did another PhD here at USF, and through all those long years of studies, I make one big commitment: no science after 8, okay? So, I'm not gonna break my rule. It's not gonna be science. It's just gonna be we're gonna talk about values. We're gonna go talk about important things tonight, okay? And here are the things. So what guys, I have children right now, so I'm thinking, what is the most valuable thing your parents can give to you? Nope. Okay, people in the front row.
Life.
Life. Good. Are you a biology student? I know you are. She's eating. She's like, oh, no. Well, I have a slide I forgot to help you, right? So what is like the most valuable stuff you can get from your parents? To close. Come on, give me insights. People in the back, I don't see much attention there. Wave to me if you can hear me. Good job. Well, no. Sorry, not that one. I'm a biologist, so I'm subjective a little bit. But really, if you think about it, the most valuable thing you can acquire from your parents is genetic information. Without genetic bingo! Genetic information. Without genetic information, we wouldn't even exist. You're right. But you didn't talk about symmetry of it, okay? So, very important. I prepared some visual aids. What is genetic information? You may think it's a toilet paper. No, it's not. This is how genetic information is. It's 6 feet of it. 6 feet. See, I'm 6 feet tall, so this is how long genetic information is, which is required in order to become human. Alright? So what is genetic information? Genetic information is a long molecule of DNA. Here you go. Gorgeous, elegant, but so long. Well, since it's so late and we don't want to be too scientific, let's act it out. I need a volunteer. Preferentially a female. All right. Applaud, guys. Beer lovers, come on. Yes, to get brave. So, how it all started? How did we acquire our genetic information? You know what I'm gonna talk about? Reproduction. All right? So, really, when you learn science, you need to visualize everything. That's why I brought this toilet paper to show you really the DNA is really long. But where it came from? Once upon a time, there was a lonely egg. What's your name?
Melanie.
What? Melanie. Or oocyte. More than anything else in her life, in her short life, only 1 day long. But in her short, so personal, right? So just acting out, act now. So in her eggish life, which was 1 day long, she really wanted to become human. She even didn't care whether she's gonna be a girl or a boy, just human. But listen, did she possess genetic information on how to become a human? Hold this. Look, how cruel. she had only a half of it. She had only a half of genetic information, and with a half of genetic information, you're not a human yet. What needed to happen? Now, the other half of genetic information need to be brought to her. Michael, do you want to be the other half? Come on. You have no choice. Audience is waiting. Ooh, applause. Once upon a time, well, she was kind of unique. He was one of many. The sperm named Michael. One day, there was so much love around, I don't know, and they accidentally completely merged. Just merged, guys, somehow. I mean, acted out somehow, I don't know. Great. Now, you remember, now, we completed. It's so noisy there, whatever it happens. Thank you, Mike. Hope they're not bored. All right. Right, listen to this. Hold this. Now she got 6 feet of genetic information. Very importantly, you think Michael was just brought her half of genetic information. He permitted her to stay alive and develop into a human. You can sit now, you're done. Well, no. Now, let's think about this. She was just one little tiny cell, even though she got all of the genetic information. Well, just for, to be entertaining, let's just look, what is genetic information? Can you read it? What does it say? It's a boy, has 2 legs, 2 arms, brown hair, blue eyes, nothing, empty, stuff like that. Biological information. Thank you so much. She got free beer from Pint of Beer and Michael too. Well, the major point I want to tell you, see, we talked about the beginning of life, and beginning of life is really to get these 6 feet of genetic information. But then, what's next? Now you still one cell. Even though you have all genetic information to be a human, nobody's gonna take you serious if you just one cell, just like nothing, right? So you need to grow. That's what we call embryogenesis, right? So what is happening during embryogenesis? That one cell starts to divide. Moving. So what we're gonna see, I don't know how many of you are biologists, but nobody saw really cells dividing. So this movie right there, showing you real time, if you don't see it, you didn't drink enough, right? That movie shows you how cells are dividing. Basically what happens, the cell grows and splits in 2. And then it grows and splits in 2, splits in 2. Sarah, slide now, we're done, right? And when you're presenting, you need to find like amazing stuff into everything. And really, that one cell after that little fertilization divided so many times that it generated our whole body, now respects you, you are visible, and your body is composed of 37, you know what it is? Trillion cells. It's so many cells that I couldn't even find you something to compare with, but the idea is if you were counting each of your cell like this, 1, 2, 3, it would take you 1 million years just to count all of your cells. Is it ridiculous? It's so many cells. Well, that was so cool. Well, let's get a little bit nerdy now, okay? We forgot about genetic information, right? So what is genetic information? It's a type of information that is really long And that information is used by our cells. So if you are made of that many cells, the previous slide, that means that each single cell in your body contains exactly the same genetic information, right? Right. So how come that each new cell in your body acquires the same genetic information? There are no more sperms around, right? At least they contained somewhere, right? So how each cell gets genetic information of its own? Well, that is because there is unique biological property of DNA. DNA as a molecule maintains itself and allows itself to be copied and then split in between two daughter cells. So really, when you watch that movie about cells dividing, what you saw, it was just a surface event. The major things happen inside of those cells, and all those cellular division was all about DNA, was all about the genetic information. There is no cell in the world that would just divide whatever it wants. It divides only if it copies its DNA first, and only if it's splitting that copied content of DNA into two cells, okay? So since it's all so complicated, scientists said, well, let's name it. And they name it the cell cycle. Okay, so what is the cell cycle? It's a very complicated event that which I don't want to talk tonight with you. And the point of that event is to make one mother cell to copy its DNA and then split that two sets of molecules of DNA between two daughter cells. Has phases, right? That's what scientists think, right? And the most important phases for us are two, S phase and M phase. So what is S phase? S phase is the time in the cell's life when cells makes copies its DNA. So, I'm so old-fashioned maybe, so I wanted you to visualize this. So how do you copy DNA? This is, see, 6 feet of DNA. Do you want to be volunteer? Sarah, you want to volunteer with me?
Come on, Ben.
Right? Oh, I get one. Thank you so much. So, what's your name? Jackie, she's going to be our S phase, and she needs 2 hands for that. So what happens during S phase? A cell, when it enters Jackie's S phase, take this molecule of DNA and makes 2 of it. How would you do that? Yep. Go ahead. Everybody, round of applause. S phase is ongoing. In real life, this takes 12 hours. So take your time. No, expedite. The faster you are, the better for evolution. Right.
Don't cut your fingers. Watch your hands.
So that's what I want you to think. When you grow, when you get more cells, it's not that you get more cells. You get more DNA. Each cell in your body has so much DNA now. If you go back here, appreciate this for a moment. I know you want to drink beer instead, but let's just appreciate it. You get 1 set of DNA. Woo-hoo! Bravo. Bravo, 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 bravo. All done. And now we have so many. Yeah, I'll take it back. But now look at this. This is the most interesting and most educational moment of the whole evening. Imagine you being a mother cell. And when you have children, you don't have favorites, right? You want to be fair. So now you have 2 molecules of DNA, 6 feet long each. And imagine it's in a tiny minuscule volume of a single cell, right? And now we need to distribute this mass between 2 daughter cells, and they're like, Mama, you need to be fair. Genetic information needs to be distributed evenly. So what do you do, Michael? How would you deal with this? Any participation? I have participation points. 10 points for participation. All right. I feel like only one table participates. Wave to me if you want to participate, but you're too shy. All right, one person in the whole hall. That's great success. All right, thank you. Do you understand my question? It's so great. Ask me, why do I teach biology? Because I have an accent. And it's such an asset because people hire me like, oh, he has an accent. It gives so many excuses to students. Well, I didn't understand the lecture, not because I didn't study, but because I didn't understand professor's accent. Make sense? Or I can say, well, excuse me, I had an accent, maybe it was not delivered efficiently. Make sense? Plus, it always gives this room for asking questions. You know, if you have an accent, students like, oh, can you repeat all of that lecture from the beginning because of your accent? So,
I understand it all. I understand everything you're saying.
yes, so let's think together. We're gonna be together now in M phase. M phase is a part of cell's life. When the cell gets this mass of thank you so much, right? You copy the DNA, there's so much of it, and now you need to separate it between two daughter cells. You know what cells do?
Multiply.
No. What do you do with DNA? They condense it. Take one end.
This one?
Whatever. Look, we're gonna be condensing DNA. See. Just roll it. I don't know. Can you start from the end so it's really organized and nicely? So why do I do this? Because I need to fill out time with something, first of all. And second of all, because I'm transitioning to another very important concept or term, a chromosome. What is a chromosome? Chromosome is nothing else but a condensed molecule of DNA, okay? So when cells divide, they cannot deal with these 6 feet, 2 molecules of each 6 feet long. It's just physically impossible. So what they do, they condense them in nice rolls like this. Let's see who can do it faster. Let's see whose roll is gonna be better. Oh my gosh, she's faster! No. Oh, gee. Well, she started before. All right, so now, look how beautiful. Hold on, see here. Now we have these two little things, and look how easy to separate them. Make sense? M phase. Oops. Now one cell gets 6 feet of genetic information, and you can keep it.
Oh, okay.
Goodbye.
I'm lying.
Well, you're gonna think, oh, that professor was so annoying. He brought paper, show us real stuff. So let's see how it really looks like. So this is how real DNA copying looks like. Ah, now you're happy, right? So this is DNA double helix. Gets pulled in some sort of special molecular machine. And from that machine, we get 2, 1, and second piece of DNA coming out. It's basically a miracle if you believe in miracles, right? So this is how it looks in real life. You know, I'm glad I brought paper, right guys? Right? So let's watch again. This movie lasts like 5 hours, so we have a lot of time. So this, what did I say, 12 hours, right? S phase lasts 12 hours. So DNA gets in, this is mother's DNA, and then 2 daughter DNAs get pulled out of it. Oh, all right. Well, I have another video, but you know, we have a video glitch today. I think somebody spilled beer on cables. But this video shows you really amazing, really nerdy situation when you have the DNA over there. Thank you, Sarah. Oh, I spent so much time looking for those videos, but it's okay. It's frustrating only to me. So that video shows you in real time how cells instead of like rolling like, very primitively, it actually rolls DNA into loops and solenoid structures and condenses it. 6 feet of DNA into tiny, super condensed and super short, importantly, piece of DNA, which is called a chromosome now. All right, well, the last slide, the most important though. Why are we talking about all genetic information? Why are we talking about genetic information if Sarah asked me to talk about cancer, right? Well, this is the situation. The DNA is a chemical. Any chemical can be chemically altered. If you alter any other chemical, not a big deal, but if you alter your genetic information, you alter what biologically wise you're supposed to be. If that happens in oocyte before you're born, then maybe you get a mutation, right? But now, see, it's still replicating. Amazing, right? But now, look at this. If you have so many cells, and each cell has molecule of DNA, has genetic information, there is so many molecules to corrupt, okay? So many molecules to alter chemically. And what alters those chemicals? There's a bunch of different substances or agents, and altogether we call them carcinogens, right? So I wanted to have another volunteer, but I'm just gonna give up on that. So what are the carcinogens? Carcinogens, again, you want to? All right, Sarah, while I'm talking, Sarah's gonna take this molecule of DNA and corrupt it. Go ahead.
Corrupt it?
Yeah, whatever. I don't know, make holes, bite it, put some ketchup on it, right? So she's gonna do all of this, tear, and look.
Sauce on it.
So
Disrespect.
you. So, listen, she corrupted this area, but let me tell you, it's irrelevant. What happened in this portion of DNA, there is no information actually. You'll be surprised, unless you're a biology student who paid attention. All these 6 feet of DNA, not all of these 6 feet of DNA contains genetic information. You'll be shocked right now. Are you ready? Only 5% of it. Now the 95% of these 6 feet, we just like, oh, we don't know what it is, satellite DNA, let's study it, all right? So only 5% of these says, I have 2 legs, I have blue eyes, all the biologically relevant information, right? So if you corrupt that information before you're born, you may not have eyes. You're most likely not going to be even born, right? But if you corrupt your genetic information throughout your life, then, well, you already have eyes, you already have legs and hair, you don't really care about those features. What you do care, you care about part of your genetic information that controls integrity of your DNA copying and DNA distribution. Because if that part of genetic information is corrupted, then DNA is not maintained properly, and that leads to cancer. Okay? So let's get practical, guys. We're drinking beer. Beer is fine, apparently. So here's some of the most popular carcinogens, the most popular ways to corrupt your DNA. Let's look. What do we have here? UV light, guys. You expose your body / your DNA to UV light, it alters it, okay? What else? The most popular, the second one, smoking, tobacco smoke. Oh, huge, so many carcinogens in it. So you smoke, you expose your cells, your DNA to tobacco molecules, they alter your DNA. The longer you live, the longer you smoke, the more chances for those molecules to corrupt very important portions of your genetic information. What else we have? Well, apple represents not fruits, pesticides. That little thing represents viruses, right? Like hepatitis virus. If hepatitis virus ruins your genetic information, And the most popular now, if you feel like deli meats, apparently not so good for you anymore. And primarily because they contain sodium nitrate and nitrates, which again are chemicals that alter chemically, that affect DNA structure and alter your genetic information. So on this really sad and dramatic and kind of down note, we're gonna just say, cheers to your DNA health, okay? Let's be conscious. Let's learn science to an extent that it's useful for us. And the most useful part of science in terms of talking about DNA biology is to value it. That's really what you are biology-wise, is based on the genetic information you acquire from your mom and dad. So say thank you for my genetic information. I'm pretty sure in the very near future we're gonna have lawyers to look over your genetic information so it's not, disclosed inappropriately and so on. But for now, that's what we are biology-wise, what we got from our parents. Each cell contains it, and if it alters biology of our cells. And in our lives, there's some diseases, some biological pathologies or cellular malfunctions that happens when our DNA is corrupted. And one of them is cancer. On this happy note, I hope I have a couple questions. If not, then I'll finish my cake, I guess. Thank you. Was inclusive, right? Cheers.
Okay, well, yes, that too. Hooray. Air cheers. Okay. What about like DNA repair? Because, we just found out that St. Pete has the highest rate of skin cancer. So, but at the same time, our bodies are fixing that, right? What's the mechanism that's involved?
Great point. A great scientific insight. The question is about a fancy term, DNA repair, right? So we all have cars and it can be repaired. Well, apparently the concept of repairing existed before we have cars. Cells have specialized molecules that allow them to detect whether DNA is intact, whether DNA is the way it's supposed to be or not. And then there is a plethora of different ways how cells can address that. So really, when you're exposed to UV light, for instance, or X-rays, and your DNA gets all messed up and ruined, so the genetic information is lost, the cells can detect that. And most likely what the cell's gonna do is gonna die. So you're gonna peel, you're gonna lose those cells, right? While in less dramatic cases, when DNA damage is not maybe so profound and massive, the cells can actually replace altered or damaged or wrong components within the molecular DNA and put the right one. So that is called DNA repair, but unfortunately we nowadays we cannot control it. We don't have drugs or therapeutic agents that can enhance your DNA repairing potential. So it's just steady at the same level, The more you expose yourself to carcinogens, your reparative mechanism may not be able to catch up with all of this. Therefore, you're going to accumulate errors. You're going to accumulate cells with altered genetic information, which can lead to cancer. Great question. So smart. Anybody else? Yes, sir.
There's been a lot recently written about telomeres. How does that fit into what you're discussing, telomere repair and telomere damage?
Oh, great question. Telomeres, another fancy stuff. I didn't know, guys, that you're so prepared. So what are the telomeres? So DNA, where's my DNA? Is a lone 6' molecule. When DNA gets copied, it's actually not so easy with scissors. The DNA kind of, goes, the machine that copies DNA cannot physically copy the whole molecule of DNA. It's going to leave ends of it uncopied. Okay? So what that means, each time a cell divides, before it divides, it copies its DNA. But every time the DNA is going to be tiny bit shorter, tiny bit shorter, tiny bit shorter, because every time those ends of DNA molecule, which are called telomeres, cannot be copied. So therefore, small quantities of DNA molecules are lost throughout your life. There is not really, as far as I know, there is not much genetic information within the telomeres, but somehow scientists think that the length of telomere indicates how long a given cell can divide. So it means how long you can live, essentially. So it's like internal clock within each cell, how long a given cell is allowed to divide before it cannot divide anymore. And therefore, if cell cannot divide, it cannot replace elderly cells in your body, and therefore you're gonna age. So that is probably, for general public, the idea would be the significance of telomeres is to limit how long a given molecule of DNA can be copied, and therefore how long a given cell can exist by dividing and keep. So.